Hi friends, today we are going to discuss about the glycogen metabolism. Here we discuss two lectures. The first one is glycogen synthesis, and the second lecture it will be glycogen catabolism, the breakdown of glycogen. So as we know that the glucose is a very essential carbohydrate required for many reactions and use as a source of energy. So here excessive amount of the glucose, it may be stored in the form of glycogen. So a readily mobilized storage form of the glucose is glycogen, which mainly synthesized in cytosol and present in the granular form. So it is a large polymer of D-glucose present in the muscles, that is 1 to 2% by weight, 400 gram, and also present in the liver, 10% by weight, that is 100 gram. It also found in the traces amount in the brain and kidney in limited amount. So here let's begin. That as we know that the glucose first convert into glucose 6-phosphate, and this glucose 6-phosphate goes different sort of reactions. Here it goes to glycolysis. Here this glucose 6-phosphate go to HMP shunt and many other several type of the reactions. But here we discuss the glycogen. So here glucose 6-phosphate is converted into glucose 1-phosphate, the replacement of glucose the phosphate group from position number 6 to position number 1. So it is isomer of each other. So we can here say that this enzyme, the work here, that is isomerase enzyme. And after the conversion of glucose 6-phosphate to glucose 1-phosphate, this glucose 1-phosphate with the help of an enzyme UDP pyrophosphorylase enzyme is working as an essentially important enzyme. It convert the glucose 6-phosphate into UDP glucose. This UDP, that is uridyl diphosphate, is added to that of the glucose, and this addition makes the glucose is reactive and is make active. And after the addition of UDP to glucose, this glucose UDP glucose is added to another compound, and this compound is called glycogenin. This glycogenin present in muscles and also in liver. Glycogenin 1 mostly found in the liver, glycogenin 2 it is mainly present in muscles, and it help in the formation of glycogen. If this glycogenin is absent, the glucose cannot convert into glycogen. So this glycogen, as we can see, that it has a tyrosine group, OH group, and this OH is replaced, this H is removed, and glucose is added to the oxygen of the glycogen by the help of an enzyme, that is tyrosine glycosyl transferase. This tyrosine glycosyl transferase then convert these two products and these two compounds into a long chain, that is glycogenin is added to glucose. Is the initial glucose is which is added to that of the glycogen. So without adding the glucose to that of the glycogen, glycogen metabolism, glycogen synthesis is not possible. So here we can see that the initial step is carried out by an enzyme, and this enzyme we discussed, that is tyrosine glycosyl transferase. So after the initial addition of the glucose to glycogenin, another essential enzyme which work here, that is glycogen synthase enzyme. This glycogen synthase enzyme is a regulatory enzyme, that is glycogen synthase. This glycogen synthase is a regulatory enzyme which, after the addition of the initial glucose to glycogenin, starts adding more and more glucose to this glycogenin. So it forms a long chain of the glucose. Added to that of the glycogen. So this long chain of a glucose is further added by the different sort of different positions by glucose. For example, this long chain is synthesized by glycogen synthase enzyme, and here addition of the glucose like this added to the different variant different side of the glucose, long chain of the glucose by an enzyme. They are called as branching enzymes. This branching enzyme is essential for the formation of the glucose 1,6 linkage and glucose 1,4 linkage. This glucose 1,6 and 1,4 glycosidic linkages help in the formation of a long polymerized chain of the glycogen, as we see here. That, for example, this is a long chain of the glucose added to the glycogen, and these branching enzymes which help in the formation of different sort of branches. So here this structure, it is what we call as glycogen. So here we discuss that is how the activation of the glycogen synthase is occur.